how allele frequencies change in a population as that population produces subsequent generations through sexual reproduction. This model left us with kind of a problem, however, and that's where I want to pick up in this lecture. Let me remind you of this difficulty. In population genetics terms, we've defined evolution as a change in the relative frequencies of alleles for a trait over time. But the Hardy-Weinberg equilibrium suggested that allele frequencies generally don't change across subsequent generations of a sexually reproducing population. What then, if anything, does this tell us about evolution? Hardy-Weinberg seemed to imply that evolution doesn't occur. Now, a resolution to this seeming paradox comes from the realization that, as is the case for any formal mathematical model, the Hardy-Weinberg equilibrium only holds if certain key assumptions are met. And generally, these assumptions are not met in natural populations. So why bother with the Hardy-Weinberg equilibrium at all? The reason is that the Hardy-Weinberg equilibrium is useful because by understanding the assumptions underlying it, biologists have been able to identify the mechanisms responsible for evolutionary change that are represented by violations of those assumptions. And in that sense, gain a broader understanding about how evolution works in populations. There are 5 key assumptions that have to be met for this Hardy-Weinberg equilibrium to hold. Let me list these assumptions first, and then we're going to look at each in more detail as we try to understand what they tell us about evolutionary mechanisms. The first assumption is that there can be no mutation in a population. That is, no new kinds of alleles arising. The second assumption is that the population must be isolated. That is, individuals can't migrate into or emigrate out of that population, and they can only breed with other individuals from the same population. The third assumption is that the population has to be large. Now, how large is large enough is a question, but this seems like a fairly straightforward assumption. The fourth assumption is that mating within the population must be random. That is, individuals can't choose whom to mate with. And the final and fifth assumption is that reproductive success must be random among individuals in the population. That is, all individuals must have an equal probability of surviving and reproducing. Now, having listed these 5 assumptions, I'm sure your first impression is that none of them are ever likely to be true in natural populations, and you'd be right. In particular, we know from the examples of natural selection we've already discussed that the last assumption, this assumption of random reproductive success, isn't going to be met if natural selection is acting on a population. Reproductive success in that case isn't random, and the differential reproductive success among individuals is what natural selection is all about. But what about the other 4 assumptions? When they're violated, how does that lead to evolutionary change? Because we know that it does. Gene frequencies will change according to the Hardy-Weinberg model if any of these assumptions are violated. So those assumptions must somehow represent a mechanism for evolutionary change. So in other words, what we're getting at is what are the other mechanisms responsible for evolutionary change other than natural selection? As we explore the answer to that question, I want you to keep in mind that what we're going to find out is that natural selection is not the sole mechanism of evolutionary change. In particular, we're going to see that a violation of the third assumption I mentioned, that is that the population has to be large, leads to a particularly important evolutionary force that we call genetic drift. Now, let's look at each of these assumptions in more detail and see what they tell us about what causes evolutionary change. The first assumption I mentioned is that mutations can't occur in populations. Obviously, from what we've learned about molecular genetics earlier in the course, we know this assumption cannot be met generally. Mutations occur spontaneously and often frequently as a result of replication errors or other sources of unrepaired damage to an organism's DNA. Mutations, as I've said, are essentially a source of new types of alleles in a population, and thus it makes sense why the Hardy-Weinberg equilibrium needs to assume that mutations don't occur. I mean, how can allele frequencies truly remain constant if entirely new alleles are being added to the mix? But the real question is, how much does the addition of new alleles really influence gene frequencies in natural populations? In other words, how strong an evolutionary force is mutation? Now, I want to clarify something, because I've said it a number of times that mutation is a very important source of variation. And this variation, the introduction of new alleles, can lead to evolutionary change. But before, when I talked about that, the implication was that those alleles might be acted on by some other evolutionary force, for example, natural selection. The question I have now is, does mutation on its own, just simply the creation of new alleles on its own, lead to significant evolutionary change in populations? And the answer to that question is generally no. Mutation will only have a direct effect on allele frequencies if there's an enormously high rate of mutation such that the allele frequencies for a single gene are all affected. Let me clarify that by giving you an example or sort of putting this in terms of how mutations might affect different genes in an organism's genome. So, for example, the human genome is estimated to have somewhere between, let's say, 40,000 to 60,000 different genes. Actually, these estimates are changing daily, but it's somewhere in that ballpark. Now, if a mutation occurs, it occurs at random, and therefore it can affect any one of those 40 to 60,000 different genes. Well, if several mutations occur, or even many mutations occur, chances are, because they're occurring at random, they'll be very unlikely to affect the same gene out of that 40 to 60,000 possible genes that could be affected. Thus, mutation rate would have to be enormous for there to be an accumulated effect for any one gene affecting the allele frequency of that gene. So the take-home message here is that mutation isn't thought to be an important evolutionary force on its own, but instead provides the source of variation on which other evolutionary mechanisms might act. Okay, let's look at this second assumption of the Hardy-Weinberg equilibrium, and that's that populations have to be isolated. What this means is that individuals can't leave or be added to a population from some other population. Now think about this for a second. When an individual moves from one population to another, what it's doing is actually carrying its alleles with it. So individuals moving across populations would represent the flow of alleles across populations. We call this in general gene flow. It makes sense that Hardy-Weinberg needs to assume that this doesn't happen, because if genes are moving in and out of a population, that could potentially change the net frequency of alleles for any particular gene. On the other hand, migration tends to be relatively random. So the likelihood that migration or immigration is going to have a significant effect on any one gene, again, like mutation, is relatively low. And so gene flow isn't thought to have a very strong effect on allele frequencies in a population. Actually, what gene flow probably most importantly does is swap alleles around between different populations, and in that sense help reduce genetic differences among populations. Okay, the third assumption of Hardy-Weinberg that we mentioned was that populations have to be large. And as I said, that unlike mutation and unlike gene flow, violation of this assumption actually is thought to be a major evolutionary force leading to significant change in populations. In fact, perhaps more of an important force than natural selection. We should ask why this matters. I mean, in other words, why should the size of a population have some sort of an effect on the way that its allele frequencies are going to change over time? So to understand this point, imagine if you would flip a coin 1,000 times. Now, I actually did this last night. Not 1,000 times, I did it 10 times, and I'll tell you the result I got in a second. But if I had done it 1,000 times, I would expect, if it's a fair coin, to get about 50% heads and 50% tails, but it wouldn't be exactly 500 heads and 500 tails. Of course it wouldn't. Sometimes it would come up the wrong way, not the wrong way, a different way. So I might get, well, let's say 530 heads and 470 tails or something like that. It would be close to 50-50, not exactly. Well, what if you flip the coin only 10 times, as I did last night? Well, what I got was 7 heads and 3 tails. Now, I think the coin was fair. Why didn't I get 5 heads and 5 tails. Actually, I would have been surprised if I got exactly 5 heads and 5 tails several times in a row, because of course it doesn't always come up exactly. The point is that the smaller the number of coin flips, the less likely you are to get a result that comes close to the predicted 50-50. This deviation is what we would call sampling error. Now let's apply this same logic to the problem of sampling from our gene pool. Imagine that the relative frequencies of 2 alleles in a gene pool is 50-50. Thus, if we reached into the gene pool at random, we would expect on average to get one allele 50% of the time and the other allele 50% of the time. If we reached in 1,000 times and pulled out 1,000 alleles, we'd probably come pretty close to an estimate of the known 50-50 ratio of alleles in that gene pool. But if we reached in only 10 times, we might get 7 of one allele and 3 of another allele. Now imagine that those 10 alleles were all that we had representing the gene pool in the next generation. We'd be starting out with a 70-30 mix, not a 50-50 mix. And as that next generation went on, it would have a very different allele frequency than the previous generation. We call this effect of sampling error on the genetic structure or gene pool of a population genetic drift, or sometimes we just call it drift for short. And it's because this sampling error can cause the allele frequencies to literally drift. I mean, in this one sampling event, they drifted by chance from 50-50 to 70-30. Natural populations are in fact relatively small. So genetic drift is thought to be a generally important evolutionary force acting on these populations. Even very large populations can sometimes be affected by drift under some circumstances. For example, at the end of the 19th century, hunters had reduced the size of the California elephant seal population to only about an estimated 20 individuals. Initially, there were thousands and thousands of these elephant seals, but because of hunting, they got reduced down to just 20 individuals. Well, the population became protected, and it quickly grew back, approaching its original size. And now, again, there are many, many California elephant seals. But their genetic makeup is very different from what the genetic makeup of the original population was. Why? Because they underwent a severe sampling error as they went through what we would call a genetic bottleneck. Another kind of event that leads to drift in populations if a small number of individuals leaves one large population and moves somewhere else, essentially colonizing a new area. This small population, small number of individuals, is very unlikely to have the same proportions of all the alleles that were represented in the original larger population. And so the gene pool of this newly established population is probably quite different from the original population because of genetic drift. We call this the founder effect. Finally, the last 2 assumptions of the Hardy-Weinberg equilibrium that I listed at the beginning are that both mating and reproductive success must be random. And of course we know that these assumptions are rarely met in natural populations. Individuals in a population are generally quite selective in which other individuals they'll mate with. And not all individuals are as successful at attracting mates as others. Thus, mating is clearly non-random. And of course, not all individuals are equally likely to survive or equally successful in producing offspring. Thus, reproductive success is not random. So if these assumptions held, it would be as though we were drawing alleles from a gene pool at random to form individuals for the next generation without any regard as to which alleles got combined or whether they got thrown into the pool for the next generation. But if there's non-random mating, it means that the alleles are not drawn at random with respect to each other. Having drawn one allele, the probability of drawing another allele will be affected. And if there's not equal reproductive success, then having drawn 2 alleles and created essentially a new individual, the likelihood that that individual will actually contribute its alleles to the next generation will depend on whether it has high or low reproductive success. Of course, we already know that natural selection is a very important evolutionary mechanism, and its violation of these last 2 assumptions that really represents what natural selection is all about. Well, let me summarize where we've gotten so far. We've seen how the formulation of the Hardy-Weinberg equilibrium identifies forces that cause evolutionary change in populations. Natural selection is one of these forces, but not the only one. In addition to selection, other forces represented by other violations of the Hardy-Weinberg equilibrium also could potentially lead to change in the genetic makeup of a population. The most important of these other evolutionary forces is genetic drift, which results from sampling error of alleles in small populations. So selection and drift are both important evolutionary forces. I want to take a second to contrast how they affect populations, because it's really in very different ways. The genetic changes in a population due to drift are not necessarily adaptive. If I'm sampling only 10 alleles and putting them into another population, I'm sampling them at random, and so whatever changes in allele frequency I get may not actually be adaptive for whatever that new population, the environment that new population finds itself in. It's just a random change, which is why we call it genetic drift. Selection, as we've seen before, actually works differently because selection leads to adaptation. In fact, selection is the only evolutionary mechanism that leads to non-random changes in allele frequencies because specifically particular alleles or combinations of alleles produce more adaptive phenotypes. That is, phenotypes that are more likely to survive and reproduce depending on whatever their genetic underpinnings are. Now, it's this change in allele frequencies in a gene pool that is due to natural selection, which provided the connection between the basic mechanisms of inheritance that Mendel discovered and the theory of natural selection as Darwin originally proposed it, that ultimately led to what I referred to in the last lecture as the modern synthesis of evolutionary biology. What's happened since is the realization that natural selection is not the sole evolutionary mechanism causing allele frequency change in populations, but just one of potentially several, and especially one that works in conjunction with genetic drift. Now, selection is one that we talk about a lot when we're talking about evolution, and the reason is because selection can lead to adaptation. It turns out that selection can affect the makeup of a population, both the genotypic and phenotypic makeup of a population, in a number of different ways, however. And I want to take a moment now to look at the different ways that natural selection can act on populations. We refer to these different ways as what we'll call different modes of selection. I just want to remind you that we're talking about one evolutionary mechanism now that leads to adaptation, and this has to be considered in terms of a suite of possible mechanisms which lead to relatively random changes. Now, remember that most complex traits, the ones that Darwin was interested in, tend to have a continuous distribution of phenotypes because they're influenced by the action of many genes. The example I gave before was the continuous distribution of skin color in humans, which results from the cumulative influence of at least 6 different alleles across 3 different genes. It turns out that these continuously distributed traits usually tend to fall along a continuum in what we would call a normal distribution, or what's more commonly called a bell-shaped curve. The bell-shaped curve in these distributions essentially is a consequence of the way that Mendelian forces act on the distribution of individuals in a sexually reproducing population, but we won't get into the details of that. Now, the point I want to make is that if we have a normal distribution, we have a particular distribution of the numbers of phenotypes with different values of the trait. The question we want to ask is what happens to this distribution if natural selection acts on the trait? The answer to this question depends, of course, on which individuals in the distribution have higher fitness than others. So 2 lectures ago, we talked about a couple of examples of natural selection which represented cases where individuals in a population at one end of a continuous distribution were better adapted than others. For example, in the Darwin's finch beak example, individuals after a drought that had larger beaks were doing better than individuals that had intermediate size or smaller beaks. So we would say that selection is favoring individuals with larger beaks. What happens to the population when selection favors one of the extremes? Well, what happens is that over time, the mean characteristic of that population will shift in the direction of the more adaptive traits. So in the finch example, over time, the distribution of beak sizes of finches shifted to larger beak sizes. This mode of selection, which is selection that causes a directional shift in the mean value of a character of a population, we call directional selection. Now, what directional selection does to a population is it changes the average value of the distribution. So if we look at our bell-shaped curve, the curve remains or can remain relatively the same shape, but it shifts to one side of the distribution or the other side, shifting to the side where the more adaptive trait is located along the continuous distribution of that trait. Directional selection is largely what Charles Darwin was talking about in his book, and it's often what we imagine when we think about evolution by natural selection, because we think of adaptation as traits becoming more of something or less of something, beaks getting bigger or beaks getting smaller. But natural selection doesn't necessarily have to have a directional effect on the characteristics of a population. In fact, evolutionary biologists have suggested that directional selection really doesn't occur very often, that it only occurs intermittently in evolutionary history during periods of environmental change, when the adaptive value of traits shift, which was the case in our Darwin's finch example. It was when the environment changed that directional selection acted on the population. Now, an entirely different way that natural selection can act on populations is what we call stabilizing selection. Stabilizing selection is what happens when the average value of a trait in a population is actually the best trait. Let me give you an example of this. A classic example comes from data that was collected around 1950 from British hospitals on birth weight in human infants. This was collected by Mary Carn and L.S. Penrose. And they looked at birth weight of over 13,000 infants, and they asked how selection might be acting on birth weight by asking whether infants of different sizes, different weights, were more or less likely to survive. And they found that there was an effect. They found that infants that were larger than average would be less likely to survive, and infants that were smaller than average would be less likely to survive. The infants that did best were the ones which were right about the average, a little over 7 pounds. Now, how is selection acting on this population? Well, the thought is that infants that are smaller than average, because they're smaller, are less well developed and therefore less able to fend for themselves physiologically when they're born. Well, you would think then that being bigger is better. But there's a very different kind of selection acting on large infants, which has to do with the average size of the human female pelvis. It's harder for larger infants to actually be born, and the stress associated with this more difficult birth is what causes selection on larger infants to act. So infants somewhere in the middle do best, and we call this stabilizing selection. Now, what's the effect of stabilizing selection on the characteristics of the population? It doesn't shift the mean character, because the mean character is the best. It's the outliers of the distribution that aren't doing as well. So the effect of stabilizing selection is actually to make this bell-shaped curve pull in its sides, to become more narrow, to reduce the total amount of variation in the population. The stronger the stabilizing selection on a population, the less variation there is around whatever mean value there is. Well, a third, again, quite different form of selection occurs when just the opposite is the case. When the mean value is somehow less adaptive, whereas the two extremes both are more adaptive. We call this kind of selection disruptive selection, or it's often called in the literature diversifying selection. Now, what happens if disruptive selection is acting on a population? What happens to the distribution of traits in that population? What we expect is over time, we would develop a bimodal distribution. This is because individuals at the mean value of the trait aren't doing as well, but individuals at the extreme are doing better. So there will be more and more individuals on both ends of the distribution over time because they are leaving more offspring, and fewer individuals in the middle. So essentially what was once an original bell-shaped curve will now essentially have two humps. It'll be a bimodal distribution. Let me give you an example of this, because this for me was a little harder to wrap my head around. How could this actually work in a natural population? Well, I'm going to give you a specific example. And this is another species of bird. This is an African bird that lives in Cameroon in West Africa called the black-bellied seed cracker, which is a great name. Like the Darwin's finches we talked about, there's an enormous amount of variation in the sizes of beaks within a population of seed crackers. And like the Darwin's finches, as the name of this species suggests, what these birds generally eat are seeds that they crack open. Now, unlike the Darwin's finches we looked at before, if you go and measure the beak sizes in a population of these seed crackers, you find that there's not a bell-shaped curve, but instead there's a clear bimodal distribution. What I mean by that is that there are two distinct peaks in the distribution. Now, there's continuous variation. You can find some birds with very, very large beaks and very, very small beaks, and you can also find a few birds that have intermediate-sized beaks, but most of the birds are have beak values that are relatively large or relatively small. So if you go to a population of black-bellied seed crackers, you'll see what appear to be more or less two very distinct kind of birds, the big-beaked birds and the little-beaked birds. Now, there's an interesting question here. What's responsible for this bimodal distribution? An ornithologist named Thomas Bates Smith, who's now at UCLA, has worked on these birds for many years. And what he found was very interesting. He found that if you measure the beak sizes of birds when they're initially born into the population, you do get a bell-shaped curve. In other words, you will find a lot of young birds who have intermediate-sized beaks. It's only when you measure the adults that you find a bimodal distribution. Now, what's happening here is that the seeds that these birds eat that are available in their environment actually fall themselves into two distinct size classes. There are a lot of very large and hard seeds, and there are a lot of very small and soft seeds. Birds with the small beaks are better at picking up and handling these small seeds and can be more efficient at eating those. Birds with the large beaks are able to crack open the larger, harder seeds. Birds that have intermediate-sized beaks are like jacks of all trades, but masters of none. They can try the small seeds and they can try the large seeds, but they're really not very good at either. So over time, actually, literally what Smith has found over the first year of life, these birds with the intermediate-sized beaks tend to not do well. They tend to not survive because they're not very good at eating. So what we have then is at the end of this is a bimodal distribution of beak sizes due to disruptive selection. Now, what this means is that the effect of disruptive selection on populations is very different again from the effects of directional selection and stabilizing selection. Directional selection shifts the mean characteristics of a population in a way that we typically associate with adaptation. Stabilizing selection reduces the variation in a population, presumably because the intermediate trait, the average value, is already the most adaptive. What disruptive selection does is actually increase genetic diversity by creating this bimodal distribution. These three modes of selection are acting in very different ways, but it's this last example of seed crackers in particular that raises an interesting question. If these birds are so strongly dimorphic, with very different large beak and small beak forms, why do we consider them to be a single species? Why don't we consider them to be two species? Well, if they are one species, will it eventually become two species? What would it take for these two forms to be considered what we'd call two species? It's these kinds of questions that we need to turn to next, and we'll begin to look at what species are and how new species arise over the course of evolution in the next lecture.